Hello everyone, and thanks for joining us for the Applications of Gene Editing 2023. I'm George Hammond, conference producer for Technology Networks, and I'm here to moderate today's event alongside my colleagues Athena Hansen-Ahern and Caroline Clark. Before we start the talks today, I just have some general information and a few tips for getting the most out of the event. Firstly, as well as a fantastic lineup of speakers we have for you, I'd like to point out the networking areas on the platform, which you can use to chat with other delegates throughout the day. On the left-hand side, you will see the People tab, where you can access a list of attendees that you can contact. From here, you can message them privately, and all your ongoing conversations will appear in the chats area. I encourage you to get involved and take this opportunity to speak to some of the over 1,000 delegates that have registered for this event. If you are not sure who you would like to chat to, we encourage you to use our participant matching feature to match with attendees who share similar interests to you. Just navigate to Participant Matching, located in the left-hand menu, and fill out a brief questionnaire. Our software will then do all the hard work for you and match you with other attendees with similar interests and goals. In the list of matches, there will also be a percentage match, which indicates how close of a match your responses were to the attendees in the list. Please remember that we encourage debate and discussion and welcome all of you to use our networking services to introduce yourselves to other attendees. But we do not permit the overt solicitation of products or services and reserve the right to block any user who infringes on this rule. On a similar note, all of today's talks offer the ability for you to engage with our speakers via the Q&A session at the end of each talk. I encourage all of you to ask questions, as this is the best opportunity to get live answers from our panel of experts. We will explain how you can ask questions at the start of each presentation. It's also incredibly important to say that this event was only made possible thanks to the generosity of our Technology Spotlight sponsor, SciEx. In the Literature and Resources section, you can download free educational pieces from our sponsors and partners, including application notes, white papers, and guides focused on gene editing technologies. For more detailed instructions on how to make the most of the features available to you at this event, please navigate to the event home and watch the overview video at the top of the page. We would like to ask if you could please refrain from video, audio, or photo recording of all talks. Many of our speakers have kindly agreed to share their slides with you, and these are available within the literature section on the platform. Some of our speakers will have included their contact details in their presentations. If they do, please feel free to reach out to them directly.
With that, I will now move on to introduce our first talk of the day. Lizard tail regeneration as an instructive model of enhanced healing capabilities in an adult amniote. I'm pleased to welcome our first speaker, Thomas Lozito, as your keynote presenter. Thomas received his BSc in Biomedical Engineering from Johns Hopkins University and his PhD from Cambridge University in stem cell biology. He joined the USC Departments of Orthopedic Surgery and Stem Cell Biology and Regenerative Medicine in 2019. Dr. Lozito's research attempts to answer the question, why can't mice regenerate tails like lizards? The Lozito lab seeks to determine the cellular and molecular determinants of the diversity in tail amputation regenerative capacities with the goal of improving an organism's natural wound healing abilities. Their 10-year goal is to create a mouse capable of spontaneously regrowing an amputated tail like a lizard. A very warm welcome to you, Thomas. Following the presentation, we'll have a Q&A session and we'll welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk. So feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during this presentation. If you experience any technical difficulties during the presentation, please request help using the technical help icon in the menu on the left-hand side of the player. Now, without further ado, I'll hand over to Thomas to begin his presentation.
Thank you very much. Thank you for this opportunity to talk about a gene editing project that we performed on regenerating lizard tails. So I'll begin with, why study lizard tails? Where lizards are amazing for a number of reasons, and many of these are due to their intermediate evolutionary position between amphibians and the salamanders. And this distinct position affects much of their biology, including their regenerative biology. So lizards are the only amniotes and closest relatives of mammals that retain enhanced regenerative capabilities as adults. Of course, The most traditional regenerative model organism is the amphibian salamander, and salamanders are able to regenerate both tails and limbs. But as amphibians, salamanders exhibit some life history aspects, some genomic architecture, and cell and tissue types that just aren't translatable to mammals. However, as amniotes, lizards retain their ability to regenerate their tails, but their close evolutionary position and relationship to mammals means that they have the same developmental processes, similar genetic architecture, and many of the same genes and cell types found in mammals, but they're still maintain this ability to regenerate an appendage as an adult. So in my lab, we focus on the lizard due to its translatable properties to mammalian regeneration. So focusing on tail regeneration, the regenerative lizard tail has some interesting properties. Here are some comparisons between salamander, lizard, and mouse tails 0 to 4 weeks following amputation. The dashed line marks the amputation plane. So while amputated mouse tails scar over, lizards and salamanders spontaneously regenerate new tails. However, if you take a look at the lizard tail, you can already start to see some differences between the regenerated and original portions of the lizard tail, while as the salamander more or less regenerates a perfect tail. We refer to the regenerated lizard tail as an imperfect regenerate because all the tissues of the tail are actually distinct from those of the original. And I'll focus mostly on the skeletons of the regenerated lizard tails. So here are, again, axolotl salamander, lizard, and mouse tails 10 weeks following amputation, analyzed by micro-CT. And while the axolotl salamander regenerated tail achieves the same patterning and segmentation in the regenerated tail, it's recapitulated similar to the original, the regenerated lizard tail is quite distinct from the vertebrae of the original tail. The regenerated lizard tail skeleton takes the form of a single unsegmented cartilage tube that is very different from the ossified vertebra of the original tail. So all of these imperfections of the imperfect lizard tail regenerate extend to the tissues of the regenerated tail. For example, the regenerated muscle is disorganized and unsegmented. Regenerated blood vessels lack valves. The spinal cord lacks new neurons. No true tendons are formed. But again, the most obvious of these imperfections concerns the regenerated lizard tail skeleton. And again, that's because the regenerated lizard tail skeleton is almost completely cartilaginous. The vertebra, the ossified vertebra of the original tail are regenerated again as a single unsegmented cartilage tube. So the vast majority of lizard cartilage resists ossification and remains as cartilage for the lifetime of the lizard. So some of these lizards can live more than 30 years and their cartilage remains as healthy, unossified cartilage for all of that time. Cartilage is a tissue that most mammals, and humans in particular, are completely unable to repair. But here's an animal that spontaneously regenerates an abundance of cartilage in response to skeletal injury. And this is actually what first attracted me to study the lizard in an orthopedic setting to determine how it's able to regenerate this robust source of cartilage. So continuing with some of this background, I'll describe the major milestones in lizard tail regeneration and cartilage tube formation. For each milestone, I'll present a sagittally sectioned representative lizard tail stained with pentachrome staining, which stains spinal cord purple, vertebra orange, cartilage green, and muscle this darker red. And above each sagittal section, I'll present crudely drawn cartoons showing the salient features of that milestone. So immediately after tail amputation or tail loss, the tail stump contains terminal vertebra and, of course, a severed spinal cord. About 7 to 10 days following tail loss, wound epidermis migrates over the stump and re-epithelializes the stump tissue. This wound epithelium thickens to what's called an apical cap. And at this time, there's an immune cell infiltration that introduces proteases to the stump environment that degrade tissue and liberate stump cells that proliferate and accumulate underneath this apical cap. These stump cells continue to proliferate to form a very important structure in the regenerative process called the blastema. This is defined as a mass of mesenchymal cells that differentiate and give rise to a majority of the regenerative tail tissues, including the cartilage tube. During this blastema stage, the regenerated spinal cord infiltrates the blastema as an ependymal tube, and this kind of nexus of crosstalk between blastema, ependymal tube, and wound epithelium drive tail regeneration, elongation, and also differentiation. And so over the next week, now a month following amputation, the cartilage tube differentiates from the blastema cells. The cartilage tube, shown here in green, completely surrounds the ependymal tube, and then you get differentiated loose connective tissue and fat around the cartilage tube, and around that is the regenerated muscle. The regenerated cartilage tube makes contact with the original tail skeleton at its proximal end. That's the attachment point between the regenerated tail skeleton and the original skeleton. So the project that I'll tell you about for this gene application compares lizard and salamander tail regeneration and asks the question, why don't lizards regenerate perfect tails like salamanders? Why do you only get this unsegmented, unpatterned cartilage tube? And to investigate this topic, we used the axolotl salamander, and we decided to use a very special lizard in these studies, the mourning gecko. This is an all-female species that exhibits parthenogenesis, or asexual clonal reproduction, where the mom lizard spontaneously lays fertilized eggs, and these eggs are viable and hatch to yield clonally identical offspring, clonally identical to each other, as well as the female that laid the eggs. This is the only lizard species that is both diploid, capable of regenerating tails, and reproduces in this manner. And this is very special because since all the offspring are genetically identical, we can take advantage of that and perform cell and tissue transplantations amongst our clonal lineages without the fear of cell or tissue rejection and without the need to use immunosuppressant drugs, which have been shown to negatively impact regeneration. So our entire colony of now over 1,000 lizards was derived from a single female. So our entire colony is clonally identical, and we can take cells and tissues and transplant them amongst any member of our colony without rejection. And since this organism is also diploid, that greatly facilitates the gene editing applications that I'll talk about in a couple minutes. Okay, so we began by comparing lizard and salamander tail cross sections. Here, immunostained with collagen 2 in green to visualize the skeletal elements, and beta-3 tubulin in red to visualize nervous tissues of both original and regenerated lizard and salamander tails. And you notice that original lizard and salamander tails are actually quite similar, with a dorsal spinal cord element and a ventral skeletal element. But when you compare this to the regenerated lizard tail, you can start to see big differences between the lizard and the original regenerated original lizard tails that you don't see in the salamander. So the regenerated salamander tails are able to recapitulate that dorsal spinal cord and ventral skeletal element, whereas the regenerated lizard tail lacks all dorsal ventral patterning. You have a tubular skeletal element completely surrounding the regenerated spinal cord element, the ependymal tube, in the center. You also start to notice big differences between the original and regenerated spinal cord. That's because the lizards don't regenerate new spinal cord neurons. Again, if you compare original lizard and salamander tails, their spinal cords are similar and both contain dorsal root ganglia, these DRGs. While lizards actually don't regenerate new neurons or DRGs, so that the regenerated lizard spinal cord takes the form of an ependymal tube, a very simplified structure compared to the spinal cord of the original. So we hypothesized that this differences in skeletal patterning and this imperfect spinal cord regeneration were linked. And this link is we've shown through hedgehog signals. So specifically, Lizard tail skeletons lack this dorsal ventral patterning because lizard regenerated spinal cord themselves lack dorsal ventral patterning of hedgehog signals. So what do I mean by that? Well, when you look at the regenerated lizard tail skeleton, its cartilage tube completely surrounds its ependymal tube. If you take a closer look at the regenerated lizard tail ependymal tube and analyze it with immunohistochemistry for sonic hedgehog, a floor plate marker, and BMP2, a roof plate marker, you can see that the regenerated lizard tail ependymal tube has sonic hedgehog expression completely surrounding its circumference. And this correlates with the cartilage that completely surrounds the ependymal tube. If you take a look at the cross-section of the regenerated salamander tail, you see a cartilage rod ventral to its ependymal tube, and analyzing the roof and floor plate markers of the salamander ependymal tube, you see that at the sonic hedgehog floor plate is restricted to the ventral side of the ependymal tube, whereas BMP2 is localized to the dorsal roof plate. So we hypothesized that this mirroring of the sonic hedgehog signal with the location of the regenerated cartilaginous skeleton was important. And we went on to do some more experiments that validated these hypotheses, and one of these involved implanting beads soaked with hedgehog modulating drugs, the hedgehog signaling inhibitor cyclopamine and the hedgehog signaling agonist SAG. So here we used beads soaked in either cyclopamine or SAG and implanted them into the either dorsal or ventral portions of lizard and salamander blastomas. So lizards implanted with control beads regenerated normal cartilage tubes that completely surrounded ependymal tubes, but the placing of a cyclopamine bead in the dorsal part of the lizard blastoma now restricted the cartilage formation to the ventral side, and the dorsal cartilage could be partially rescued by co-implanting a cyclopamine bead with a SAG bead, the agonist, to rescue that cartilage formation. And very similar story in the salamander tails. Salamanders, again, normally regenerate a cartilage rod ventral to the ependymal tube. Placing a cyclopamine bead now in the ventral blastoma completely inhibits all cartilage form differentiation. And this can be partially rescued by co-implantation with a SAG bead. So these results strengthened and suggested to us that it was hedgehog signaling in both lizards and salamanders that was inducing cartilage formation. And we hypothesized that the regenerated lizard tail skeleton lacked this dorsal ventral patterning in their cartilage because the regenerated lizard ependymal tube lacks dorsal ventral patterning signals, and that introducing dorsalizing and or suppression of ventralizing signals in the lizard ependymal tube will restrict these pattern skeletons to form a pattern more similar to that of the salamander. So our question became, why do lizard ependymal tubes lack these dorsalizing signals? Well, it turns out this is due to populations of neurostem cells within the spinal cords of both salamander and lizards. So both salamander and lizard spinal cords contain populations of neurostem cells that express the marker SOX2 and culture as neurospheres. So when you take these neurostem cells out of the tails of these animals, they self-aggregate into these structures called neurospheres. Now, the salamander spinal cord again contains different domains, PAC7 positive roof plate, a PAC6 positive lateral domain, and a sonic hedgehog positive floor plate. And these SOX2 positive neurostem cells are found in all of these domains, throughout these domains in the salamander spinal cord. But if you look at the lizard spinal cord, again, the lizard lacks the roof plate and lateral domains. It's entirely composed of sonic hedgehog positive floor plate, completely surrounding its circumference. And the lizard spinal cord also is made up of these SOX2 positive neurostem cells. But again, these are all sonic hedgehog positive floor plate identity. So we again extracted the neurostem cells from these two sets of spinal cords, cultured them as neurospheres, and then asked what the neurosphere neurostem cells expressed in terms of floor, lateral, and roof plate domain identity. And we found something very interesting. When the salamander neurostem cells are taken out and cultured as neurospheres, they default towards a PAX7 positive roof plate domain. They lose their PAX6 and sonic hedgehog positive identities and more or less become a uniform PAX7 positive population. And then conversely, the lizard, when you take out its neurostem cells and culture as neurospheres, they retain their sonic hedgehog positive floor plate identity. So this was our first major difference detected between salamander and lizard neurostem cells. And we'd like to say that the salamander neurostem cells default towards a roof plate identity, while as the lizard neurostem cells default towards a floor plate identity. And we think this has major implications in how the regenerated tails are patterned between these two organisms. We also detected differences in the effects of sonic hedgehog signaling on these neurostem cells in vivo and in vitro. So the first set of experiments will be in live animals in vitro. And again, under control conditions, you get dorsal ventral pattern skeleton with collagen 2 now stained in this white color, this grayish white color. Also, under control conditions, you have a dorsal ventrally patterned salamander ependymal tube with sonic hedgehog positive floor plate and a PAX7 positive roof plate. And again, under control conditions, the lizard lacks dorsal ventral patterning in both its skeleton and its ependymal tube. Treating systemically with the hedgehog inhibitor cyclopamine inhibits cartilage formation in both salamander and lizard, again, because lizard blastema cells respond to hedgehog signaling with chondrogenesis. So inhibiting hedgehog signaling will inhibit cartilage formation in both of these animals. But when you look at the ependymal tube in salamander, cyclopamine treatment leads to an expansion of PAX7 positive roof plate at the expense of floor plate, whereas lizards are not affected by cyclopamine. They maintain their floor plate. And then conversely, treating the lizards and salamanders with the hedgehog agonist SAG, now you get an expansion of sonic hedgehog positive floor plate and in the salamander, but again, you don't see really any difference or changes in the salamander ependymal tube. Also noticed in SAG-treated animals, you get a massive expansion of chondrogenesis in the blastemas of both salamander and lizards. So these results showed us that these drugs are definitely affecting the hedgehog signaling environment in both the salamander and lizard blastemas. We know this because it's very much affecting the chondrogenesis in these animals, but only the salamander ependymal tube was responsive to these modulations in hedgehog signaling. The lizard was stubborn and just maintained its sonic hedgehog positive floor plate despite these drug treatments. We confirmed these findings in vitro by treating our neurospheres, again, with cyclopamine and SAG to modulate hedgehog signaling in vitro. And now the salamander neurostem cells, again, take on a roof plate identity typified by PAX7 positive staining. Treatment with cyclopamine, they maintained this roof plate identity, but treatment with hedgehog agonist SAG switched the salamander neurostem cells to a sonic hedgehog positive floor plate identity. Whereas then the lizard, the neurostem cells maintain floor plate identity despite any of these treatments. So again, salamander neurostem cells were responsive to hedgehog manipulation, which with SAG and hedgehog and agonism ventralizing the salamander neurostem cells, just like we saw in vivo, whereas the lizard just maintained their floor plate identity no matter what the treatment. We also compare the differentiation capacities of salamander and lizard neurosphere neurostem cells in vitro. We found that the salamander neurostem cells readily differentiated into neurons, shown in this white color, under these differentiation conditions, whereas the lizard neurostem cells were unable to differentiate into neurons and just recapitulated a glial phenotype under differentiation conditions. So in summary of this first part of my talk, we detected differences in salamander and lizard neurostem cell populations. Salamander neurostem cells are extracted from distinct roof plate, lateral domain, and floor plate domains of the original spinal cord. In culture, they default towards a roof plate, multipotent neurostem cell that, depending on the hedgehog signaling environment, differentiate into roof, lateral, and floor plate domains in the regenerated salamander spinal cord. Whereas the lizard story is much more restricted. The original spinal cord is entirely made up of floor plate identity. When you take these neurostem cells out of this environment, they maintain their floor plate identity and exhibit a restricted differentiation capabilities that keep them as floor plate. There's lots of sonic hedgehog signaling within the regenerated tail, so all the environment is, again, pushing them towards floor plate. So what you end up with is a regenerated lizard spinal cord that is completely floor plate. So going back to our goals and our hypothesis, we somehow needed to manipulate the lizard neurostem cell populations within the regenerating lizard tail to enhance roof plate identity and suppress sonic hedgehog signaling in order to get the patterned ependymal tube and hence the patterned regenerated tail skeleton in regenerated tails. So this is the kind of plan that we sought to seek out. And on this, in these attempts to phenocopy the salamander dorsal ventral patterning, we turn to a transplantation system in which we take neurostem cells, again, from either salamander and lizard spinal cord, culture them as neurospheres, label these neurospheres with a vital dye, fluorescent dye, dye-I, and then implant these neurospheres back into the ependymal tubes of these live animals, where they engraft and merge with the native ependymal tube, and then are recapitulated in the regenerated tail. So what that looks like is, again, we're amputating these tails, injecting dye-I labeled neurospheres into the ependyma of these amputated tail spinal cords. The dye-I labeled populations contribute and recapitulate a portion of the regenerated tail ependyma, so that when the tail regenerates, you have these exogenous manipulated neurostem cells within the regenerated tail. So here's a sagittal section of a regenerated lizard tail spinal cord showing the neurostem cell populations in SOX2 and our exogenous cell populations in dye-I. And you can see that the exogenous neurostem cells contribute and recapitulate portions of the regenerated ependymal tube. When we compare, when we analyze these exogenous neurostem cells for either beta-3 tubulin as a marker of neurogenesis or glial filamentary accessory protein as an indicator of gliogenesis, we see that the exogenous neurostem cells do not undergo neurogenesis, but they do contribute to the gliogenesis of the regenerated tail. So we performed this interesting experiment where now we took neurostem cells from both salamander and lizard tails, labeled them with the dye-I label, and then injected and transplanted these neurostem cells into lizard spinal cord ependyma. So now we have lizard with exogenous salamander neurostem cells and as control exogenous lizard neurostem cells to see if we can copy the lizard tail regeneration dorsal ventral patterning by introducing salamander neurostem cells now into the lizard. We analyzed the cross sections of the ependymal tube for our PAX7 positive roof plate marker and sonic hedgehog, our floor plate marker, and then we treated the animals with either vehicle control or cyclopamine. So these are all lizards, and now these lizards received salamander neurostem cells. Under control conditions, the dye labeled exogenous cells persisted, but without the shielding from the exogenous hedgehog signaling environment. They lost their PAX7 positive phenotype and were completely converted to sonic hedgehog positive floor plate. However, when we protected them from the sonic hedgehog signaling environment within regenerated lizard tails, they persisted and maintained their PAX7 positive roof plate identity. And for the first time, we started to see lizard tails with patterned ependyma, with a PAX7 positive roof plate and a sonic hedgehog positive floor plate through the introduction of salamander neurostem cells. But again, only when we treated with cyclopamine to protect them from the high native endogenous hedgehog signaling environment. And then, of course, our control lizard neurostem cells did exactly what you would expect. They never expressed the PAX7 positive roof plate domain. They maintain their sonic hedgehog positive floor plate domain, and the regenerative ependymal tubes were no different from untreated animals. So our plan became this. In order to phenocopy the regenerated lizard tail, we not only had to introduce neurostem cell populations capable of roof plate identity, but we needed to protect those exogenous neurostem cells from the high and rampant hedgehog signaling present within the regenerated lizard tail. So we needed to find a roof plate capable neurostem cell population and then make that population resistant to hedgehog signaling. So again, our goals became twofold: to identify a lizard neurostem cell source that exhibits roof plate identity, and to engineer roof plate neurostem cells that do not ventralize in response to hedgehog signaling. Following this plan, this is what we hypothesized that would result in not only dorsal ventrally patterned ependyma, but also finally dorsal ventrally patterned skeleton. So we found our roof plate competent neurostem cells in embryonic neural tube, the embryonic neural tube of embryonic lizards. These contain SOX2 positive neurostem cell populations, quite similar to the adult salamander and lizard spinal cords. And again, these were culturable as neurospheres. So just like in the salamander, the embryonic lizard spinal cord contains PAX7 positive roof plate, PAX6 lateral domain, and sonic hedgehog positive floor plate, with SOX2 positive neurostem cells found throughout these domains. And just like in the salamander, when you culture these embryonic neurostem cells as neurospheres, they default towards a PAX7 positive floor plate domain. So basically, the embryonic lizard neurostem cells were quite similar to the adult salamander neurostem cells and exhibited many of the same properties. And finally, we have now a lizard cell source that's able to give us our roof plate identity. And again, the adult lizard neurostem cells again default towards a sonic hedgehog positive floor plate. Also, very much like the salamander neurostem cells, the embryonic lizard neurostem cells are able to differentiate into neurons, where again, as the adult lizard neurostem cells cannot. So our strategy here focused on making hedgehog unresponsive embryonic lizard neurostem cells. The canonical hedgehog signaling pathway passes through a switch molecule called SMO. SMO acts as a gatekeeper for the hedgehog signaling, and if SMO is inhibited or knocked out, the result is our cells that are unresponsive to hedgehog signaling. So we focused on knocking out SMO in lizard embryonic neurostem cells. We use the CRISPR-Cas9 gene editing technology to introduce double-strand breaks in the lizard SMO gene that stochastically would result in indels that would knock out the lizard SMO gene expression. So our strategy employed SMO knockout plasmid containing a cassette for Cas9 expression, as well as a guide RNA designed to target the lizard SMO gene, as well as a homology directed repair plasmid with complementary left and right flanking arms and a GFP insert in the middle. The idea being that the combination of these two plasmids in the lizard neurostem cells would create a double-strand break and basically replace the lizard SMO gene with a GFP gene, allowing us to flow cytometry and FACS sort the successfully edited green fluorescent cells that had their SMO genes knocked out. So our strategy here was to isolate embryonic neural tube cells, expose them to the Cas9 gene editing system with a SMO-directed guide RNA and a GFP HDR plasmid, creating a mixed population of edited and unedited cells, isolate and enrich for the edited neural tube cells, culture these as neurospheres, implant them into the ependema of adult lizards, and then study the effects on the patterning in the regenerated lizard tail. So using this strategy, we successfully generated GFP expressing edited embryonic lizard neurostem cells in a high efficiency, and these cells expressed significant and almost complete knockdown of the SMO RNA, indicative of an unresponsive hedgehog phenotype. And when we implanted these edited neurostem cells into the adult lizard ependema, I'm happy to report that we finally saw the first regenerative lizard tails with patterned skeletons and spinal cords. So in these cross sections of our lizards treated with the SMO CRISPR edited embryonic neural stem cells, here the cartilage is visualized in red. For the first time, we saw cartilage restricted to the ventrum of these tails, similar to the cartilage rods that you see in salamanders. Taking a closer look, we also saw DRG-like structures in these edited stem cell treated animals that expressed the neural marker TUJ1, as well as our GFP marker, indicating that they were derived from our implanted edited stem cells. Again, these stem cells are able to differentiate into neurons, unlike the adult stem cells, and this resulted in neurogenesis within the regenerated lizard tails. And taking a look at the ependema of these edited neurostem cell treated animals, we saw exactly what we expected. The GFP tagged, the DII and GFP tagged exogenous edited neurostem cells expressed PAX7 in these tails. Sonic hedgehog was now restricted to the floor plate, and finally we got this patterning between PAX7 in the dorsal part of the ependymal tube and Sonic hedgehog in the ventral part. And we believe that this is what was able to recapitulate this dorsal ventral patterning in these lizard tails. And then the control tails that received control CRISPR neurostem cells, again, just exhibited the control cartilage neural tube that completely surrounded the ependymal tube and an unpatterned neural tube. So in conclusion, not only did these edited neurostem cells result in the first new neurons in regenerated lizard tails, but also form the first patterned skeletons and also ependymal tubes in these animals. So our hypothesis and our pathway towards phenocopying the salamander regeneration lizard proved to be successful by enhancing roof-flight identity and simultaneously suppressing hedgehog signaling in these animals. We created nature's first regenerative lizard tails with dorsal ventral pattern skeleton and ependymal tube. So with that, I'd like to conclude this presentation of this gene editing application to alter the fidelity of a regenerative organism, in this case, the regenerating lizard tail. I'd like to acknowledge the lab and the people who performed this work, specifically Megan Hudnall, a very talented research technician that did a lot of the transplantation studies that really allowed us to test our model in live animals, as well as our funding sources from the Commonwealth of Pennsylvania, as well as the NIH R01 that funded this work from NIGMS. And I'll happily take any questions or comments on this work.
Thank you very much, Thomas. That was such a fascinating talk to kick the day off with.
you.
We're likely to have lots of questions from the audience, and I can see that we've received some already, so I'll move straight on to those now.
Sure.
So the first question you have is, how many times can lizards regenerate their tails?
Right. So we performed experiments where we re-amputate regenerated lizard tails within the regenerated portion. So like a re-regenerate, if you will. And all the lizard species that we tested so far can actually re-regenerate their tails a number of times, as long as the lizard is healthy and below a certain age. Once the lizard gets a little bit too old, we do see a reduction in its ability to regenerate its tail, and this transfers to its ability to re-regenerate tails. But the regenerative process is robust enough that it can start with either original tail tissue or regenerative tail tissue, form a blastema, and regenerate a number of times.
That's great. Thank you for that. I'll move on to your second question, and that is, do regenerated lizard tails function similar to the originals?
Right, that's an excellent question. It actually depends on the species and the main functions of the tail. For example, some lizards, the main function of the tail is as fat storage. Lizards don't store a lot of fat on their bodies, but they store fat in their tails, and this fat is an important caloric reserve during the months of the year where the lizard's diet of insects might not be available. So for those lizard species that fat storage is the most important job of the tail, their regenerated tail is structured in a way that it's very good at storing fat, but it loses its ability to move more or less. You get calcification of the cartilage tube, but an expansion of the fat reserves in the tail. Because in those lizard species, the tail is used as fat storage, a tail that's not able to move as much might not affect its survival. But some lizard species use their tail for climbing, and we have some of those species in the lab as well. And we find that those lizard species, when they regenerate their tails, are able to resist that calcification. So their regenerated tail is kept as dexterous and as flexible as the original, and they're still able to use their regenerated tail as a climbing assist. So in terms of just, within the lizard group, there's different lizard species that are able to modulate the end product of the regenerative process, so that their regenerated tail is as functional and as useful as that specific lizard species uses in the wild. And that's another avenue of study, is how these lizard species are actually able to, affect the regenerative outcomes of their tails, something that I find really fascinating.
It certainly is fascinating. And moving on to your next question, and it is, can all lizards regenerate their tails?
Right. So that's another good question. No, not all lizard species can regenerate their tails. So it's believed, based on the fossil record, that the very first lizards to evolve did maintain and carry over their tail regenerative processes from the amphibians that they evolved from. But over the course of the millennia and the speciation of the different lizard groups, certain lizard groups lost their ability to regenerate their tails. And something that we're interested in my lab is looking at these different lizard groups to see if these regenerative lizard regenerative capability losses occurred due to the same molecular reasons. For example, there's one group of lizards, the agamids, that lost their ability to regenerate their tail relatively early in evolution. So this is an old lizard group that historically and ancestrally can't regenerate their tails. But then there's certain lizard gecko groups that have lost their ability to regenerate relatively recently in history. So we're comparing the molecular signatures of these different lizard groups to see if early regenerative tail loss versus late regenerative tail loss, evolutionarily speaking, occur due to the similar genes or other factors affecting regenerative outcome. Yeah, not all lizards can regenerate their tails, but we believe that the different lizard groups that have lost their ability to regenerate their tails might have lost that ability due to different molecular reasons.
That's great. Thank you, Thomas. And we've got time for one more question. Does age affect lizard tail regenerative abilities?
So that's a great question. Age oftentimes does affect the healing responses of animals, and we see a drop off in regenerative capabilities in most animal species as they age. We see something similar in the lizard, but for most of the adult life of the lizard, the ability to regenerate arises from its first birthday, right after hatching. Hatchlings can regenerate their tails, all the way up to old adults can regenerate their tails. However, if there's any health or condition deterioration, we do see that reflected in their regenerative capabilities, but that's not always linked to age. It's more the general health of the animal. If if the animal becomes sick or exhausted from having to keep regenerating its tail, then we see a reduction in its regenerative capabilities. But generally, the lizards are able to maintain their regenerative capabilities throughout their entire lives.
Thank you so much, Thomas. Such a fascinating topic. But unfortunately, that's all we've got time for today. Any questions that we didn't have time to get to will be sent to Thomas after the event. And as with all the other talks in the event, this talk will now be made to view whenever you want for the next 30 days. That just leaves me to say thanks again, Thomas, and we will be back soon with the next talk.
Thank you very much.